Hi everyone, I'm Genevieve. I live in Australia on Bunurong land as part of the Kulin Nation. Today I'm going to talk to you about scaling up science. This is something I care a lot about because I'm a scientist and a programmer, so I spend a lot of my time thinking about how we can use computer programming to accelerate and be a force multiplier for scientific discovery. Specifically, I want to talk to you today about Dask. Dask is a flexible library for parallel computing in Python. It has two parts: dynamic task scheduling and collections optimized for big data and parallel processing. They run on top of the task scheduling engine. Collections include parallel arrays, data frames, and lists, which we call bags. By mirroring the API of familiar scientific libraries like pandas and NumPy, it makes it easier for researchers and data scientists to use as a drop-in replacement. Dask Distributed lets you scale up from your laptop to a cluster without making changes to your code. This talk has two parts. First, we're going to take a tour through several different areas of science. We'll look at how Dask is being used in a variety of scientific Python libraries. Second, I'm going to talk a bit about how you might integrate Dask into an existing codebase. By the end of this talk, I hope you'll see that Dask is used in a lot more places than you might originally expect, and you'll have some tips for how you might go about integrating Dask with your own work. So we're going to touch on a wide variety of scientific fields for this tour and spend a short time on each. The first stop on our tour is the field of statistical genetics. SGKit is a general purpose toolkit for quantitative and population genetics. Now, this is a field with extremely large datasets. It's not uncommon to see data from hundreds of thousands of individuals with millions of variant markers genome-wide. Each variant can be thought of as a position on the genome, and each sample is taken from an individual person. That means the data can be represented in tables like this, where each row is a genetic variant and each column represents a single individual. Now, Dask is used fairly extensively in SGKit. It's used in several different ways. First, it's used for lazy loading of input data, and then Dask array is used in conjunction with xarray as a data container structure. Handling some of the computational heavy lifting is DaskML, Dask's machine learning library. We see this especially for tasks like principal components analysis. Because SGKit has this matrix data format, there's a lot of linear algebra involved, and it makes really heavy use of singular value decomposition with Dask. Last, the Dask data frame collection also gets a bit of a workout, although because this is in connection only with specific file formats, it's technically optional to use. Tom White recently gave this great short talk at the Dask Summit in May this year. It focuses particularly on some of the current challenges they have when working with large-scale computation for genomics. Tom's one of the developers of SGKit, and he's an excellent speaker, so if you're interested in diving deeper into this topic, I really recommend to check it out. Next up is single-cell analysis. Conventional bulk sequencing techniques take genetic material extracted from large numbers of cells. Single-cell sequencing, like the name suggests, takes genetic sequence information only from individual cells. SciPy is a library that does analysis and visualization of spatial molecular data. One really common application here are studies into cell differentiation. Because changes in the RNA drive the differentiation process, single-cell analysis is really well matched to this type of problem. Single-cell analysis means that we don't just have genetic sequence information. Instead, we have this plus spatial information about where that cell came from within a tissue sample. Cross-referencing spatial information and those features back with the sequencing data gives us a much richer, more detailed picture of what's going on. The newest release of SciPy, version 1.1, has just been released. And it includes a bunch of cool stuff, like support for image Z-stacks, more tutorials on image segmentation tools, like we have the animation up here. This animation is showing us some highlights from a segmentation workflow. Notably, the newest SciPy release also includes upgrades to the image container class, which now supports Dask. Now, this isn't the only recent pull request in SquidPy involving Dask, but it is the one I'm going to focus on and highlight here today. It integrates Dask image with the SquidPy image container class. Dask image is a specialist image processing library. It's built on top of Dask and SciPy to provide image-specific functionality. Michael, the author of this pull request, has put together this example using the new functionality in SquidPy, and shown here on the right is the result of a watershed operation. Watershedding is a really common image processing task, which is used to split segmented objects that are touching one another. So here they used about a 10 pixel overlap in this case, and that was enough to avoid unwanted artifacts between the edges of those image chunks. Really early benchmarking of performance for this latest change showed a factor of 2 speed improvement for this particular example. On a single machine with 8 CPU cores, the time taken for this example dropped from 20 seconds down to 10 seconds. So that's a pretty solid win, even on this 8-core machine, so that's likely a fairly standard laptop. It means that they've effectively removed this ceiling limit for computation as well, because it's going to be much easier to scale up onto a compute cluster, which could probably provide even bigger performance gains. Switching gears now to some very different types of datasets and applications, next up is data visualization and image processing with the Nipari library. Nipari is a multi-dimensional image viewer. You can see a screenshot on the right here of the main Nipari interface. There's the main viewing area, which in this example has a picture of some fluorescent cell nuclei. Up in the corner there, in this brighter region, you can see a cell undergoing mitosis, so it's dividing in two. And on the left-hand sidebar are the most commonly used Nipari controls. In the top left, you can control the display of your data, changing things like opacity, the contrast limits, and the color map. Below that is the Nipari layer list. So you can open multiple datasets and display them simultaneously. But Nipari isn't a standalone image viewer. The real power here is that it's written in Python, so you can use it interactively. It can be launched from a Jupyter notebook or integrated with a Python script. That means you have seamless access to anything in the scientific Python universe, and it allows you to get a much tighter feedback loop when you're trying out new ideas. So Nipari handles Dask arrays natively. This is very powerful for multidimensional data because it means that we can do lazy loading and processing of image data. If you put your data into a Dask array, Nipari will know how to handle that. On top of it, Nipari has this plugin system, so it's really easy to extend with additional functionality. The animation that we have playing right now is a demonstration of how Nipari and Dask can work together really well. It was put together by Tally Lambert, and it's taking data from a lattice light sheet microscope. And because of the way these microscopes are set up with the optics, the raw data that's being saved from the lattice light sheet microscope is going to be skewed or deformed. So that means there's a few really interesting steps that need to happen here. We need to lazily load the raw data from disk into a Dask array. And then a computation needs to be applied to deskew the raw data, and that's happening with Dask's map blocks. Finally, we need to run another computation like before, but this time it's to deconvolve the data to make it less blurry and kind of sharpen the image that we see on the screen. So that turns the skewed and slightly blurry raw data into something that's well aligned, crisp and clear. And this processing is happening entirely on demand and on the fly, just for the time points that are being selected. So if you'd like to learn a bit more in detail about how exactly this example was put together, I really recommend looking at the tutorial Tally has written about using Nipari and Dask together. You can find that on the Nipari website at this link. For another example of Dask being used very effectively, we can look at the Beetle Safari package by Robert Hayes. So this data that we're looking at here is of a red flower beetle in very early stages of its development. And what they're doing is using Dask not only to load data lazily into Nipari, but also to do a lot of the image processing work as well. So in the animation, we can see that there's some spot detection happening. And Then also, after the spots are detected, there's segmentation around each cell nuclei. Now, the last library I'm going to talk about on our whirlwind tour is Xarray. Xarray supports labeling of arrays with dimensions and coordinates. If you've been paying close attention, you might have noticed that we've seen Xarray pop up in a whole bunch of different contexts in this talk already. We saw Xarray used for statistical genetics as the basis of the dataset object in SGKit. We see Xarray again in SciPy, particularly in connection with the image container class that I talked about earlier. And finally, with Nipari, while it doesn't currently support Xarray natively, there are some discussions happening about that behind the scenes. So that's why I thought it would be really important to highlight Xarray. Having access to good, flexible data structures enables so many other libraries in so many other areas of science. It doesn't matter if they're working in very different scientific fields, very different spaces with data that looks and feels very different from one another, because Xarray is really quite a flexible concept. It works well in all of these different situations. So all of the examples I've shown you so far in this talk have been focused around different areas of life science, but we also see Xarray being used not just for those areas of science, but also for other areas of bioinformatics, astronomy, physics, but it's perhaps most well known for in context with geospatial data. So here are a couple of more typical examples that you might see with Xarray. On the left, there's raster image data overlaid onto a satellite imagery, and on the right, we can see the projected air temperature data from a winter and a summer happening in 2013 over North America. If you're interested in geospatial data in Dask, I really recommend checking out the Pangeo project and website for more examples and more details there. Now, if you're using Xarray, Dask is an entirely optional extra. But if you do want to take advantage of that, then what happens is that the Dask array collection is used as a drop-in replacement to chunk up these multi-dimensional arrays. So that's been a very quick overview of Dask being used by libraries in several different areas of science. We've looked at statistical genetics, single-cell analysis, and spatial omics, as well as visualization and image processing. Now we're going to move on and show you how you might integrate Dask into an existing code base. The first thing to consider is the 80-20 rule. Now, originally, this rule was an observation about economics describing wealth distribution in Italy. And while the exact ratio is up for debate, it's been surprisingly effective when generalized to other contexts. So the general rule is going to be that something like 20% of the effort will produce 80% of the results. By being strategic and doing this type of analysis on where to focus your energy, you can aim to get more bang for your buck, at least initially while you're rolling out functionality. So once you've done some thinking around that, it's time to drill down into a specific user story. You've likely identified that the bulk of your users are taking some very similar journeys, so you might want to prioritize that. The other factor to consider is that you want this user story ideally to be something that is not easy to do today. And finally, it's really important that the user story be an end-to-end workflow. There's no point in beefing up only part of your code and finding out that your users can't complete a full task. With that in place, you're ready to start prototyping your code. You're going to take that end-to-end workflow of the user story and start using that to base your prototype on. It's really helpful to work with a scaled down dataset. This reduces the feedback loop when you're iterating on a process. That makes your development much faster overall. Don't fall into the trap of thinking that just because you're trying to add support for large data, it means that you need to always be working with these large, unwieldy datasets. Pick something small. There is no minimum size for a Dask data frame, array, or bag. You can choose something absolutely tiny and only increase the size later on when you actually need to test that specifically. Now, in this process, it's going to make the pain points and the areas of Dask incompatibility with your current code very clear to you. So next, you have to go through each of those pain points one by one and fix them. Now, at this point, you're probably thinking that this advice might sound remarkably similar to the advice given here about how to draw an owl. And, well, that's because it's really hard to give good general advice, because it's going to depend so much on your specific example. I don't want to generalize things so much that all that we have left are platitudes, so I'm not going to do that. Instead, I'm going to show you a specific example of something I've worked through, and we'll talk through that. And then afterwards, I'll take you through some of the Dask best practices as guidelines. They are specific, actionable tips that you should have at the top of your mind when you're doing this type of work. In the interest of time, the specific example I'll walk you through will be pretty brief, but I have a blog post that explains all of this in much more detail, as well as a Jupyter notebook with example code. So the worked example we're going to look at today is to do with a library called Scan. Scan is written by Juan Nunes Iglesias, and it's a library for skeleton network analysis. Skeleton shapes appear in all sorts of sciencey problems, like the branching of blood vessels or airways, root structures in plants, capillaries in leaves, even like the neuron network structure in brain tissue. So there's lots of cool applications for this type of work. And Scan is accelerated by number, so it's actually quite a performant library. One thing it doesn't do well is, however, deal with very large input images. Data that has lots and lots of voxels can run you into problems with memory errors if you have constrained RAM. And that's the problem that we're going to try and solve here with Dask. So this is the basic workflow that we're trying to optimize. We've thought about our 80-20 rule and come up with a good user story, and this is the end-to-end process that we're expecting a typical user will take. So with our example, a user has a large 3D set of image data, and inside it is a skeleton-like object. They're going to need to segment the skeleton from a background and then thin that down so that the input is something that Scan will recognize. Then, with Scan, we're going to compute the branch statistics and give back to our scientists something that they can analyze for their paper. Working through the prototype uncovered a number of problems. Some, like the first one here, were expected. The others, not so much. Let's look at each of these in turn. So the first problem was the one we'd anticipated. We already knew that large input data, images with lots and lots of voxels, can run into memory problems when you use scikit image and skeletonized function. To solve this, I did two things. First, I rechunked the image data so that the chunks matched a much more efficient chunking for the skeletonized function. The image data that I'd been reading in from disk went into my Dask array with a single chunk equal to each 2D image plane. But we already know that the skeletonized function will need a 3D block, so it's going to make a lot more sense if I make my Dask array chunks match that 3D block shape as well. There is a cost associated with rechunking like this, but in this particular case, I thought that the benefits definitely outweighed the downside. And the second thing I did to solve this was to run the skeletonized function using Dask's map overlap. Now, so long as I've picked a reasonable chunk size, I shouldn't be running out of RAM anymore. It took a little bit of experimenting to find a good chunk size. That's something that's going to depend on the kinds of computational resources that you have available. So the second problem popped up pretty soon in Scan. We need to look at each part of the image to identify the different parts of the skeleton, but different parts of the Dask array will return different amounts of data, and some of the chunks won't have any part of the skeleton in that region of image at all. So it turns out that this pattern doesn't really comfortably fit with map overlap like I used to solve the previous issue. Map overlap is doing just too many clever things to concatenate the results back together, and it means that I needed to take a different angle. The solution here was to use Dask delayed and write a custom function. Dask delayed has no restrictions on what functions can return. It can be any arbitrary object. The downside is that we have to handle concatenating all the results together, but that part is at least relatively straightforward to do. One big performance tip for you is to consider passing the meta keyword argument whenever you're creating Dask data frames. If you don't give this information to Dask, it's pretty clever and it will go off and try and work it out on its own. But that can be pretty inefficient. In my case, I was creating data frames inside a list comprehension, and so this was like exceedingly inefficient, and because as a user I know in this particular case what Dask should expect in terms of the column types, it made things a lot more efficient and straightforward to be able to give that information up front. So that made a huge difference. The third problem is that since we're now using delayed instead of map overlap, it means we need to find a way to grab the array chunk with some overlap attached to it. So delayed is really flexible, which is great, but it does mean that we have to handle a lot of these like associated stuff ourselves. So the solution here turned out to be pretty straightforward. I found a very similar kind of function internally used in Dask, and I was able to adapt it to solve this issue. The last problem comes at the very last stage, summarizing the branch statistics in Scan. So Scan was designed with a handy skeleton object class to consolidate a lot of information about image skeletons together. But it turns out that some of the Dask incompatible parts of Scan were getting called in the initialization function. So for the proof of concept work, I created a temporary workaround, initializing an object instance with very small data and then systematically overriding all of the attributes with the real values. For the more permanent fix, I now have a really clear target to modify a specific section of the Scan code.
Dask has a number of best practices to keep in mind. The first rule of Dask is don't use Dask. If you can reduce your data down or make some kind of sampling from it, using other libraries like Pandas or NumPy is a much better way to go. If you absolutely have to use Dask, that's fine, but you should also stop using Dask as soon as you can and switch back if your data happens to be condensed down into something small enough where that's an option again. If the reason you're looking at Dask is because you want to speed up your code, it's important to profile it first and see if you can optimize it in other ways. Also, if you want to increase the speed, maybe consider compiling your code with Python or Number instead. The next tip is to use the Dask dashboard. It's incredibly helpful to see what's going on and is really useful for identifying like areas of inefficiency. With Dask computations themselves, you want to avoid very large partitions or chunks, and similarly, you want to avoid having very large task graphs with an excessive number of nodes. Finding your Goldilocks regions will depend a little bit on what kind of computing resources you have available, but it's not too hard to find something reasonable. It's also really important to be storing large data in a smart way on disk. It'll really affect how easily you can access it. And for that, we need efficient file formats. For data frames, we recommend Parquet. For arrays, we recommend ZAR. Dask website also has specific pages for arrays and data frame tips. The only thing I'm going to mention here is that for arrays, you should really think carefully about how to align and orient your chunks well with the computation. And for data frames, you really want to avoid any full data shuffles wherever possible. That's something that's really going to make it quite inefficient compared to other operations. Dask delayed can sometimes be a little bit harder to get your head around. So we'll just walk through some of the tips from the best practices document we have here. Now the big one is that you want to call delayed on the function itself, not the result that's returned from the function. If you try and delay the result by mistake, the function is going to run immediately before we reach the delay, and that's the opposite of what we want. Mutating the inputs of functions is also bad, as is relying on global state. These things might work well in an environment with serial execution, but once you start doing distributed computation, things will start to break. You can't rely on those assumptions, so you want to avoid it. You want to break up delayed computations, not bundle it all into just one. Now, the reason for that is every delayed call becomes one node in the Dask task graph. So if you want any advantage from parallelization, you have to have more than one so they can be sent off to different workers simultaneously. And on the flip side of that, you also don't want too many delayed calls, because that's bad too. Doing things with Dask always introduces some amount of overhead. It's like sending somebody to bring you back a pile of rocks one pebble at a time instead of bringing a bucket. Now, you never want to call delayed from within another delayed function. This is a recipe for confusion, especially confusing error messages. Finally, you also shouldn't call delayed on collections like Dask arrays or Dask data frames. And that's because collections in Dask are already optimized for that purpose, so you'll probably just wind up slowing things down. Okay, if you're going to take a picture, this is the slide to screenshot. Today we've gone on a tour to see how Dask is used under the hood in a range of different scientific computing libraries. We've seen examples from statistical genetics, single-cell analysis, and spatial omics, right through to microscopy, visualization, and image analysis. I've also given you some starting points for how you might go about integrating Dask into your own code. I hope that it's helpful for you if you're thinking about getting stuck in and experimenting. I'm always happy to answer questions, and you can find me anytime on Twitter and on the Dask Slack channel.